So let's hear more about the specifics of how this actually works. So Mary, thank you again.
Right, this is called Insta-Gene matrix. It's one type of a purifying reagent, and if you look carefully at the bottom of this bottle, you can see it's white. And the reason it's white is that Insta-Gene matrix contains little beads which capture and remove inhibitory ions from the saliva and from the cheek cells. Now, you're going to need to dispense 200 microliters, 0.2 of a mil, of Insta-Gene matrix into screw cap tubes. And if you see these screw cap tubes down the bottom of the tube, you can see the little beads that do the purifying.
Okay, now, so Mary, can I ask, do we need to shake this up before we extract, yes?
Yeah. Now to actually dispense the Insta-Gene matrix, you need to give the bottle a really good shake. And you then need to measure out 0.2 of a mil into each tube. And I find this is a useful way to do it. This is a 1 mil syringe, piece of silicone tubing, and I've attached it to, I attach it to a glass pasta pipette. John, would you mind giving that bottle another shake for me, please?
It sediments quickly, doesn't it?
Yeah.
Okay.
If you could take the cap off for me as well, thank you. I'm lazy here.
Yeah, have an assistant without using them.
Yeah, and if you just draw up to 0.2 on the syringe, and 0.2 of a mil, 200 microliters, goes into the
tube.
Have
the lid back on there in a second, and this screwed. Mary, I think you mentioned earlier to me about why we now need these screw tops as opposed to the flip tops.
Yeah, what we're going to do after we put our DNA into this screw cap tube is we're going to heat the cells to get the DNA out, and if we had flip top tubes, the
pressure could just pop it open,
just pop it open and we'd lose our sample.
Yeah, so back to square one.
So we need to have screw cap tubes. Now, what we're now going to do is we're going to put 20 microliters of our cells.
We'll bring those over for you, that. And your preference, do we do saliva cells or cheek cells? Because we don't need to do all four for the camera, we just pick one.
No, that's right. I don't mind which one.
Well, I'm going to pick the first one I come to in that corner, and according to that, those are cheek cells.
Okay, yep. So I'm just going to mix the Insta-Gene matrix a bit before I add the cells. Take the cap off.
Okay,
and I've set my micropipette to 20 microliters.
Yeah. So yes, another example of the good practice in rehearsing pipette use to start the session off.
Yeah. And then...
Do you want to hold that, or should I hold that?
Thank you very much.
Okay.
And I'm just going to take up 20 microliters of cells. And I'm just going to place them into the Insta-Gene matrix. And if you pipette up and down a couple of times, it does mix the cells.
Okay, so actually within the solution itself, yeah.
Make sure the plunger is right down to the hard stop before you remove the tip, and hold the
so last thing is
right
when
down.
Taking any of our valuable cells out, yes, okay.
And afterwards, discard the tip into Virkon, because it does contain biological material.
Okay, very good. And clearly we're just going to repeat that for the other three samples.
If you could label that tube.
I will indeed, if I have a pen.
There's one over.
Thank you very much.
Very important that it's that one, John. It's that one.
It's that one?
Yeah. Label it with, that's it then. That's
Okay.
your cheek cells and
So it's
your
got
name.
JB and C for cheek cells, and on the top it's got IG for Insta-Gene matrix.
Yeah.